A warm welcome, everyone, to the 12th edition of the Infogest Viva webinars on food digestion. We are hosting you here from Ireland, Chagask, and today my co-host here is Eva, and we are joined by Andrea Rivera del Rio, all the way from Mexico. She had to wake up very early for us today. Andrea, we are glad to have you. And currently, Andrea is working at the National Autonomous University of Mexico. She completed her PhD at the Food Process Engineering Group in Wageningen. And today she's going to present her topic, plant proteins from processing to digestion. It's going to be in a recorded format, but after the recording, she is going to be here to answer your questions. So if you do have any questions, pop them in the Q&A below, and she'll be happy to reply to those questions. So for now, we will pay attention to her talk.
Hello. Today I will speak to you about my PhD project, which was conducted at Wageningen University at the Food Process Engineering Group under the supervision of Anja Jansen and Remco Boon. But before I start, I would like to thank the Chagas postdoc team for their very kind invitation and for giving me the opportunity to speak to you today. So as I was saying, I will talk to you about my PhD project, which was entitled Plant Proteins from Processing to Digestion. I'm sure it will come as no news to you that we are in a much urgent need for a protein transition, one that goes from foods that are environmentally impactful to those that can be more sustainable. And one example of that are plant proteins. However, we rarely consume plant proteins as they are in their raw state. We usually process them either immediately into a finished product or to an intermediate ingredient that will then be incorporated into a finished product that we can eat. And of course, the goal of that product is to nourish us and for us to be able to obtain and assimilate the proteins in them, especially considering that proteins are the critical macronutrient for food security. So we need to also understand and the impact or the importance of how digestible they can be to achieve to really make a sustainable protein transition. So these were all the motivations behind this PhD project. And essentially, we followed two main roads. One that went about plant protein processing and its relationship to digestion, and another one in which we studied the dynamic aspects of protein digestion and their importance on the overall digestion. We started with a review to understand what we knew thus far about the impact of processing on the digestion, the in vitro digestion of different crops from that were sources to plant proteins. Then we conducted a study in which we studied specifically spray-dried protein isolates, and we studied the effect of heat treatment on their gastric digestion. And it is at this moment that we started walking down the path of the dynamic protein digestion. Here, what we did was study trypsin kinetics, but actually what the impact of a dynamic gastric digestion was on on the kinetics of trypsin. Then we came sort of at the intersection of these two roads, where we did a study on mild aqueous fractionation of pea protein, but we also used the semi-dynamic in vitro digestion method. So here we combine the two aspects or these two roads that I'm talking to you about. Then with the results of this study, we conducted a life cycle impact assessment, what we call a farm to feces approach. And this is sort of where we kind of crystallized all our insights on the effect of processing on protein digestion into the sustainability assessment. And lastly, what we did walking down the dynamic digestion protein path, we created an in silico model to simulate protein digestion. So the key takeaways from my PhD were mainly four. We learned that processing history affects plant protein in vitro digestibility. And throughout this presentation, I will show you a number of ways in which this happens. We learned that protein digestibility should be included in the life cycle assessment of ingredients and finished products. We, on the other side, on the other path that I'm talking about, we learned that the gastric phase influences the efficiency of small intestinal protein digestion, and in fact actually improves it. And lastly, we learned that we can simulate dynamic aspects of protein digestion with an in silico model. So on this first study that we did, this review, these were basically the main results out of them. We found a number of studies that were conducted, which you can see here, and also that they were studied under for a number of different crops, as you can see here. And in this table, if you see a red dot, it means that that the effect of this specific processing step was negative on this specific crop. If you have a green dot, it means that the effect was positive, and if you have a blue dot, it means that the effect was neutral, or that there was a net zero effect. And if you see a dash, it means that there has, at the time that we did this review, there had been no studies reported on combining or studying the effect of a specific processing step on that specific crop. Well, actually, the effect on digestion, I should say. So the first thing that is kind of evident from this table is how empty it is, sort of. There are some crops that are more heavily studied than others, and there are some processing steps that are more heavily studied than others, or more thoroughly, rather. So the other, so what we know is that maybe what we could think is, okay, maybe we don't need to study every processing step for every crop, but maybe we do need to do that, because if you take, for example, dry fractionation as one example of a processing step that was studied for a number of crops, you can see that, for example, for yellow pea, it has a negative effect, whereas for quinoa, it has a positive effect, and for lentil, the effect is neutral. So here we could say that dry fractionation is not impacting the digestibility of these crops in the same way. Then you have examples such as alkaline treatment, in which the effect is almost always negative. We have an exception on quinoa, but here we should mention that the importance of conditions, of course. The conditions for these processing steps could all be very different. So the effect perhaps for quinoa was just not as intensely shown because the conditions were a bit milder. So this is also something that is important. So of course, when we say processing, it can mean a number of different things, not only because you have different processing steps, but because you can also have different conditions. And also when we talk about different crops or plant proteins as a whole cannot mean one single entity. There are quite some nuances if we talk about different crops, and then furthermore if we talk about fractions from these different crops. So yes, so that was kind of where we stood at the time. I should also briefly mention that, of course, the digestion measurements were different to different extents among studies. It could be that we saw the report of digestion as a degree of hydrolysis, or TCA soluble protein, or free amino acids. They were all rather different, but you can find more details about this in this review paper. Then on the second study that we did, here I want to show you some light microscopy observations of spray-dried soy protein isolate that was just dispersed in water. So what we saw was that the particles that were created during spray drying were actually conserved in the dispersion, and we could actually observe them under the microscope. And here I am pointing at this specific particle that is sort of slightly attached to this larger cluster of several particles. And what we did was we added drops of simulated gastric fluid directly onto the slide, and we observed what happened under the microscope. And this is what we saw over a range of roughly 7 minutes. As you can see, this single particle becomes detached from the larger cluster and slowly degrades and diffuses onto the background. We can no longer see it at the end of 7 minutes. And the cluster also starts to erode from outside in, and it also degrades significantly toward the end of the 7 minutes. So here what we saw was that an untreated soy protein isolate could actually be evidently hydrolyzed or degraded under the microscope, as well as other determinations we did. But then when we contrast that to observations of heat-treated soy protein isolate dispersions, we see some aggregates being formed, so really on a micro scale, but the aggregation does take place. And when we do the same thing of adding the simulated gastric fluid directly onto the slide, we actually see no significant changes happening microscopically. There are some changes that we could measure via degree of hydrolysis or size exclusion chromatography, but the difference between or what occurs after heat treatment is rather evident. Right, so at this moment I would like to now shift gears and go now walking down the other road that I was talking to you about, the dynamic protein digestion road. And I want you to think about us when we are fasted. We have a small amount of gastric fluid in the stomach, and as soon as we eat, we consume a meal, the food bolus enters the stomach, and it is at that moment that gastric emptying starts from the stomach onto the small intestine. And so the chyme that is first being emptied from the stomach has been exposed to virtually no gastric digestion. And as the gastric phase continues, more gastric fluid is secreted. The bolus or the digesta is exposed to the acid environment and is subject to pepsin-catalyzed hydrolysis, and the chyme that is emptied then from the stomach now has been exposed to a complete gastric hydrolysis. So this is sort of what I want you to think about when I say dynamic gastric digestion. And what we did is that we studied the kinetics of trypsin-catalyzed hydrolysis using isothermal titration calorimetry of bovine serum albumin. We here now have changed from plant proteins to an animal protein because here we needed more model protein, especially one that was very soluble, that was more fitting for the ITC, the isothermal titration calorimetry method. And so what we had was an intact neutral BSA. So this one would simulate the chyme that is first emptied from the stomach, and a pepsin hydrolysid. That one would be the one that would be last emptied from the stomach into the small intestine. And just to understand what the role of acid is besides pepsin activation, we also included an intact acidified bovine serum albumin. And these are the results we got from the study. We first measured a hydrolysis ratio by trypsin onto these 3 different substrates. And what we saw was that the pepsin hydrolysid was the most extensively hydrolyzed substrate by trypsin compared to the intact neutral. So just to remind you again, this would be the protein that is first emptied from the stomach. In principle, this one would be more limitedly hydrolyzed by trypsin compared to a pepsin hydrolysid. And then the intact acidified is slightly higher than the intact neutral, but nowhere near the pepsin hydrolysid. And then what we now actually, with the ITC, what we did with the isothermal titration calorimetry, we determine trypsin kinetics. And what you can see here with the catalytic constants is that the pepsin hydrolysis has much higher efficiency compared to the intact neutral substrate. And here, this is why we say that the gastric phase or gastric digestion ensures that the trypsin, or well, in this case, trypsin hydrolysis taking place in the small intestine can be much more efficient. And why does this happen? Well, what we did was study or determine the tryptophan fluorescence. BSA has two tryptophan residues, mainly at its core. And what we learned was that there was a pH-induced conformational isomerization. Essentially, what happens is that at neutral to alkaline pH, BSA has a tighter structure. And then in an acid denatured state, which is only slightly reversible, so this is important because we first have an acid pH in the stomach, and then we have a more neutral pH in the small intestine. So this is why we studied the reversibility of this. This is only slightly reversible. And so acid, the pH induces a conformational state that is more, that presents a better accessibility for trypsin. And then, of course, we also have the aspect of the hydrolysis history. So not only the pH, but also the hydrolysis that took place in the stomach. This also affects how accessible or exposed the cleavage sites are to trypsin catalyzed hydrolysis. And if you ask me, this is quite remarkable because if you think about it, the gastric phase is rather brief compared to the small intestinal phase, and yet it has such a big impact on how efficient and extensive it's going to be in the small intestine. And luckily for us, at the time, the Infogest standardized semi-dynamic in vitro digestion method came out, and we were very eager to use it for our next study because we had already learned and appreciated how important the gastric phase, the dynamic aspects of the gastric phase were. So if you're not familiar with the semi-dynamic in vitro digestion method, I will briefly walk you through it. The start is very similar to the Infogest static method. You have a food that is subjected to the oral phase with a simulated salivary fluid, either with amylase or without. Then the gastric phase starts, and what happens here and differs from the static method is that the simulated gastric fluid with acid is dosed continually onto the gastric vessel, as well as pepsin. This is done separately to prevent excessive autolysis from pepsin. And what you can see is that at the beginning of the gastric phase, the pH is higher, and then toward the end of the gastric phase, the concentration of pepsin is higher then. And then after that, we have 5 gastric emptying steps that are designed to simulate the in vitro gastric emptying rate of 2 kilocalories per minute. And then each one of these gastric emptying steps is followed by a small intestinal phase. In our study, we conducted the small intestinal phase with pancreatin. And so this is sort of the data, or one type of data that you can get out of this semi-dynamic in vitro digestion method. We first have the gastric phase with its 5 gastric emptying steps. Here, what we did was measure the degree of hydrolysis. And then each one of these gastric emptying steps is followed by a small intestinal digestion that is done over 120 minutes. And so you have, yes, we took aliquots at 30 and 60 minutes of the small intestinal digestion. So this is the data that we get out of. At the end, you have these 5 points for the small intestinal digestion over 120 minutes. And what I want to draw your attention to is the fact that we have this range, of course, after this 120 minutes of small intestinal digestion. We have as a difference between the first and the last gastric emptying point of 33%. And of course, this difference comes partly from how extensively the proteins are hydrolyzed in the stomach. And what you can see here is that the difference between the first and the last gastric emptying step in the gastric phase is of 15%. So we still have to account for roughly 18% degree of hydrolysis that is different in the small intestinal phase. And we studied what the effect of protein to enzyme ratio was, because of course that changes, but that wasn't impacting significantly on the degree of hydrolysis. So here, this recalls what, or makes us remember what we learned in the previous study about the efficiency and extent of protein digestion in the small intestine because of the gastric phase. So here, once more, we can say that the dynamic aspects of gastric digestion, or that the gastric phase improves the efficiency and extent, in this case, of the small intestinal digestion or hydrolysis of protein there. And since we're talking about gastric acid or the gastric phase, it is much more than pepsin activation and protein hydrolysis. It is also important for the release and uptake of minerals and vitamin B12, etc., among other important aspects of the gastric phase. So again, very important, yet seemingly so minimal in the overall digestion. Right, so this is the point where I was saying that we are sort of at the intersection of the two roads, because we combine the dynamic aspects of protein digestion with plant protein processing and digestion, specifically yellow pea. Here we compared the conventional to fractionation method to a mild aqueous fractionation method. So first I want to walk you through the conventional method. We obtain a flour from the crop or the seed, and then we conduct an alkaline extraction, then an isoelectric precipitation. We centrifuge between these two steps, and then what we obtain, we spray dry, and we can get a protein isolate. With the mild aqueous fractionation, what we do is, after we obtain the flour, we steep just in water this flour, and then we do a centrifugation step. And here is where we can separate a protein-rich fraction, which is the supernatant, from a starch-rich fraction, the pellet. After a second centrifugation step, we can separate the soluble from the non-soluble protein fraction. So these four, the protein isolate and the mild fractions, is what we used to study their digestion with the semi-dynamic in vitro digestion method. And what we also wanted to study was the effect of processing post-fractionation. So what we did was heat treat dispersions of these fractions at 90 degrees Celsius for 5 minutes. So here we can compare the two different fractionation steps, but also processing post-fractionation. And one more thing that I should say is that if you notice here in the mild aqueous fractionation, we do not have a drying step. So we can actually say that these proteins are in their native state, which we confirmed this with DSC. So here we have the results for the, again, degree of hydrolysis for the 5 gastric emptying steps in the gastric phase and in the small intestinal phase, at the end of the small intestinal phase, of the mild pea protein fractions. Again, these are native proteins. And what we can see is that there is actually a difference between these, the degree of hydrolysis that can be achieved for these 3 different fractions, the soluble protein fraction being significantly more extensively hydrolyzed than the other 2. And so here, this makes us think that protein structure and solubility as a result of that is important for plant protein digestibility. Then when we compare that to a pea protein isolate, to the pea protein isolate unheated, this one, of course, was not heated with this post-fractionation treatment, but it had been subjected to heat treatment during spray drying. So here we know that the proteins are denatured. And what you can see is that during the gastric phase, we couldn't really see much difference between the isolate and the soluble protein fraction. But then during the small intestinal phase, once more the soluble protein fraction was more extensively hydrolyzed. So here we are not thinking only in terms of denaturation. We from the previous slide, we learned that structure and solubility were important. And here denaturation might not have such an positive effect as one might think. When we heat treated the mild fractions, here you have the protein-rich fraction after the small intestinal phase was finished, and we heat treated. So the hollow symbols are the heat-treated dispersions, and the solid symbols are the unheated dispersions. We see that because of heat treatment, the degree of hydrolysis that can be achieved increases. This also happens for the non-soluble phase, for the non-soluble fraction, although to a lesser extent. Interestingly, though, for the soluble protein fraction, the degree of hydrolysis decreases as a result of heat treatment. So here we can say that protein denaturation does not always improve digestibility if we express that as degree of hydrolysis. And for the isolate, when we heat treated it, so these are the hollow squares, we actually couldn't really see a significant difference, although there was a slight decrease of extent of hydrolysis for the last gastric emptying steps. And lastly, what I would like to do is compare the mild fractions, the heat-treated mild fractions, to the pea protein isolate when it's unheated to when it is heated. And what you can see for the unheated one is that we still have a slightly higher degrees of hydrolysis for the mild fractions, specifically for the soluble protein fraction. But this actually is much more evident when we heat treat the pea protein isolate. The protein-rich fraction and the non-soluble protein fractions are more extensively hydrolyzed. Still, when the degree of hydrolysis was lower for the soluble protein fraction after heat treatment compared to its unheated counterpart. So this is for us was interesting to see, because here's where we said we could probably make a call for as minimally processed as possible ingredients, so that when they are further processed into a final product, they can preserve some of a structure that is better suited for enzyme accessibility and enzyme catalyzed hydrolysis. So this was roughly the idea behind these results, that they sort of make a case for these mild fractionation methods. And as I said, we wanted to extend this toward the sustainability conversation, and we conducted a life cycle impact assessment. Usually, life cycle impact assessments are conducted in what we like to call a farm-to-fork approach, which comes from you get the raw material, you transform it into an ingredient, and then into a food product. However, we wanted to extend it in what we have coined a farm-to-feces approach that extends this beyond just the production of the food product into the protein digestibility of that food product, and later on into how digestible the indispensable amino acids in that product are. We use the recipe method, which here you have quite some information, but essentially what I want to convey to you is that from all of these different impact categories, we can reach a normalized single score that represents the total environmental impact. So this gives us a way to more easily compare different processes, different origins of foods into one single score. And so first I want to show you the product-oriented metrics. Here you have the single score reported for the functional unit per kilogram of protein, so the environmental impact per kilogram of ingredient. You have the mild fractions, the protein-rich fraction, soluble protein fraction, non-soluble protein fraction, and the pea protein isolate. And you also have them when they are unheated and heated as dispersions. So here, of course, you can imagine that you can expect that the mild fractions have a much lower environmental impact because they do not use resources or chemicals to change the pH. And more importantly, we did not use a drying step. So the energy and resources required to produce these fractions is much lower than the pea protein isolate. But then to compare this a bit more fairly, we have the functional unit per kilogram of protein. And here the comparison, yes, it's a bit more just because we take into account that these are very diluted compared to the pea protein isolate that is rather has a very high purity. But still, when we consider that, we can still say that these fractions, the mild fractions, have a much lower environmental impact compared to the pea protein isolate. Okay, much lower was maybe exaggerating, but it is lower. But as I was saying, we want to take this down to to consider the in vitro digestion. So this figure I showed you before, it's the degree of hydrolysis of specifically the pea protein isolate. And what we did was take the last small intestinal measurement, and we took an average of these 5 points. And we consider this the hydrolyzable protein, or the protein that can be hydrolyzed. And the reason behind doing this and why we are keen on the semi-dynamic digestion method is because all of these happen. All of this is true. The chyme is emptied from the stomach as soon as the gastric phase starts, but also it can stay until the end of the gastric phase. So all of these degrees of hydrolysis are possible. This, I should say, is regarding only luminal small intestinal digestion. We are still yet to understand or being able to determine what happens when we consider also brush border hydrolysis. So right, we took this hydrolyzable protein as an average of the degree of hydrolysis of the last small intestinal aliquot for the 5 gastric emptying steps. And this is what we incorporated here. Here you have the functional unit per kilogram of hydrolyzable protein for the mild fractions and for the pea protein isolate when the dispersions were unheated and heat treated. And here is where everything gets more interesting, in my opinion, because yes, the pea protein isolate has still higher environmental impact, but here is where the processing history for fractionation, of course, but also post-fractionation can be more easily appreciated. If you remember, the pea protein rich fraction degree of hydrolysis was higher when the dispersion was heat treated. And this is what you see here. You have a lower environmental impact for producing the same amount of protein that can be hydrolyzed after the small luminal small intestinal phase. And this was not the case for the pea soluble protein fraction, where the digestibility was lower, or the degree of hydrolysis was lower, and this can also be appreciated here. So with this, what we want to say is that, okay, so the goal of these protein-rich products is to nourish us, and we should be able to take up the proteins in them. Okay, so then we can actually express the environmental impact of these, considering how digestible the proteins are. For good measure, we also compared our results to a whey protein isolate. We did the life cycle assessment for the whey protein isolate as well as the semi-dynamic in vitro digestion. And well, to be expected that the impact is higher. But once more, we can still appreciate and include into the sustainability assessment the impact of the processing history onto the digestion of the protein. So with this, I would like to wrap up on the protein digestion part, but we actually, to continue on to the dynamic protein digestion, we created an in silico protein digestion model in which specifically we used the model to compare solid, liquid, and blended meals. And we described a number of different aspects of digestion in the model. But luckily for me, I've had another opportunity to speak to the network in a webinar a couple of years ago, and here is where I presented our in silico model. So if you're interested in that, please feel free to go ahead and look at this video or to search for our paper on this. Right, so today we have talked about how processing history affects plant protein in vitro digestibility. We showed how this happens in a number of ways. We showed you how this can be included into the life cycle assessment of ingredients and finished products. And we talked a bit at length how the gastric phase influences the efficiency and actually improves the efficiency of small intestinal protein digestion. And we briefly mentioned the in silico model that we did. So I would like to still take a couple of minutes to speak to you about my current role. I am very happy and proud to be back at my home university in Mexico as an assistant professor at the Department of Food and Biotechnology at the School of Chemistry from the National Autonomous University of Mexico, UNAM. And as I was making this slide, here you have a picture of our School of Chemistry. But as I was making this slide, I was reminded of our university crest that has this little piece of the map that includes all of Latin America. And it made me think about the efforts that Infogest is taking toward extending the network to Latin America. And I am very interested in keep working on digestion research. So to anyone over here in Latin America that would like to reach out and collaborate, I am more than happy to discuss ways in which we can do that. So my lines of research over here at UNAM, we are continuing with the in silico model. We are extending it to include spatial and temporal pH changes in the stomach. But we are still very interested in the protein transition. So now we are collaborating with ETH in Zurich in studying microalgae and the effect of processing history that it can have on in vitro digestion. And we have another project that we are exploring. It's very much in its, we are exploring at the moment how we can include the processing history and in vitro digestion of Mexican diets and incorporate that into a sustainability assessment. This is far less straightforward than just considering a single source of protein. This is diets as a whole, but so therefore that is why we are just exploring this at the moment. And yes, so of course here once more the Infogest network has a very important role. So again, anyone interested in reaching out to collaborate in these projects or any projects that you may have, I am very much eager to hear about them. And so with this, I would like to wrap up and thank you very much for your attention, and also acknowledge everyone that has contributed to this work that I presented to you today. Thank you so much to everyone.
Thank you very much, Andrea. Congratulations for your very nice presentation. So now we are starting with the Q&A section. That actually we have some question here. And the first is, the in vitro studies are interesting work. However, in reality, the body's physiology adjusts to the secretion of pancreatin to the available amount of still digestible proteins. How would that affect the interpretation?
Yes, thank you. So here are a couple of things that this question makes me think about. So first of all, indeed, it's true. The pancreas, our digestive system is incredibly efficient and compensates in very clever ways. However, with pancreatin or with this in vitro work and what we know about proteases is that they have a specificity, right? So even if we had more pancreatin or proteases there, the degree of hydrolysis would have a limit because these proteins or these enzymes rather only can hydrolyze certain cleavage sites. That said, there is a bridge that we still need to cross that is what happens between the luminal digestion and then the brush border enzyme hydrolysis, right? The catalyzed hydrolysis. So here, this is still something that we do not fully understand yet, how the degree of hydrolysis that we can expect in the luminal digestion will actually impact the brush borders. Like it could be the case that it almost doesn't matter if a protein is less extensively hydrolyzed in the luminal phase, but once it reaches the brush borders, exoproteases, they will hydrolyze and most of the protein can be taken up. So this is something that we still don't know. But yeah, so indeed. And then the other thing I wanted to say about pancreatin or the pancreas response could be regarding, for example, trypsin inhibitors. It has been reported that if you have a significant presence of trypsin or chymotrypsin inhibitors, the pancreas will also respond by releasing more enzymes. However, these can, in the long term, but I want to emphasize this quite a bit, the long term is important, that will have an impact on the pancreatic health as well. So yes, it could compensate, but that could also come at a cost.
Okay, thanks, Andrea. So we are going for the second question. Is what happens to undigested and unabsorbed proteins and its fractions? Will they enter the colon and be fermented?
Yes. So again, we still need to know what happens at the brush border. So that is whatever is not digested after that, indeed, it enters the colon. And here we actually do not want that much protein, that much intact protein to enter the colon. Some of it is necessary. Some peptides are necessary to have proliferation of the beneficial microbiota. We do want that. But to an extensive extent, then this could make, this will make very happy sulfur-reducing bacteria. They would have more to feast on. And then these bacteria can release some harmful compounds from their metabolism that we also don't want. So this is, again, a bit of a balancing act, and with still the big question mark around brush border enzymes.
Yes. Okay, we have a third question, maybe it's related to this. So will people compensate in their diet for undigested protein, e.g. eat more?
That's definitely a possibility. We have a bit both sides of the coin, right? We do have people that would probably have no issue with that because they already overconsume or consume more protein than they need. So if some of it is undigestible, then there is enough to compensate for that. But we are a bit, when we think about the protein transition, we think about kind of a very
there might be a scarcity, and then this would be, I mean, maybe I am going too much in the future, but maybe not so much if we consider, yes, everything around the use of resources. And yes, however, yes, so like as I was saying, the other side of the coin is that you do not consume enough, and then even in that case, or you consume just enough for the requirement, but if some of that is not digestible, then that might be an issue, right? Yeah.
Okay, thanks, Andrea. So we have another question here. Is nice presentation. This message is from Marcos Infantes. For the life cycle assessment, I think, analysis, perhaps the comparison between meal fraction and protein isolate was not completely fair because a stabilization step was not included for the meal fractions. These fractions still contain a high moisture content, so short shelf life and need either a heat treatment and or drying step to be commercially viable. What would the impact of this extra step on the LCA analysis?
Yes, thanks. Well, yeah, so here there are a couple of things that we thought about. If you think about a product that might have a high moisture content, so a final product that might have a high moisture content, then it almost seems that you could cut the intermediary dry ingredient, right? Because you produce the ingredient, you fractionate, you obtain a concentrate, and then you dry it, or a concentrate or an isolate. Let's talk about both at the same time. You then dry it, and then, of course, that is useful in a number of ways. That is very convenient. But then what happens is you then hydrate it again, and then you process it further. And a bit the idea that we were coming from when we did the semi-dynamic in vitro digestion method, we noticed that we sort of came up with like an inverted U shape in which you do want some degree of, some extent of processing. You do want some processing to go on with your protein to make it more available, to make it more digestible. But at a certain point, you will probably reach somewhat of a maximum. And after that, that will come down. So probably more extensive processing will again come at a cost to the digestibility of proteins. With that in mind, we thought, okay, so maybe what we could aim for is less extensively processed ingredient that will then be further processed as a finished product. And then this will have closer to this maximum digestibility possible and not further down. So it's true, in the LCIA, we are comparing wet ingredients to dry ingredients. So that's why this first functional unit per kilogram of protein that I showed you was so, the difference was very, very stark. But then when we compare it, that's why the functional units come at hand. Then we have the heating step, right? This is where we think that we will stabilize the mild fractions. I'm talking about more in a microbiological way. If we could have any sort of effect from that part, then this heat treatment could serve sort of as a pasteurization step. And then this could represent not only the processing, the post-fractionation processing, but also what could make this ingredient more stable. So what we are proposing is a more inline process that goes from the fractionation to the processing into the finished product without having to have this drying step in the middle. But yes, it's true. This is kind of a bit of a change in the paradigm that we think of, that we, the way we think that we can produce final products. We usually come from a raw material, then to an ingredient, and then combine a number of ingredients into a finished products. But this is a possibility, right? That we can come up with.
Okay, thanks, Andrea. We have two more questions here. This is from Barbara Riga. Thank you. Concerning the fractionation process, what do you think about the dry fractionation?
Yes, well, I think it's again a very good alternative for using less resources. Yes, so a less environmentally impactful process. We did not study dry fractionation ourselves, but we did find a number of studies, as I showed you with the review, that studied the dry fractionation and then the effect of that on digestibility. Once more, I think that the post-fractionation process, so with dry fractionation, we can expect that the proteins will still be native because we do not have a heating step or any other treatment that might alter the proteins. So probably there they are native. That can have its own implications on techno-functionality, of course. We do not know if these native proteins are attractive for their application in food products. Maybe they do need a heat treatment or any kind of treatment to become more functional. But again, they will, this gives us an idea that maybe once they are further processed into a finished product, then their digestibility or the degree of hydrolysis that we can obtain. I'm using these two terms a bit interchangeably, but let's say degree of hydrolysis, it could be, yeah, it could be interesting, of course.
Okay, thank you. We have another. So this question is from Patricia Duque Estrada. I was wondering about the effect of trypsin inhibitors on protein digestibility, considering the different levels of processing. Did you also investigate trypsin inhibitor activity?
Yes, we did. Hi, Patty. Yes, we did do it in the pea protein study. We did not get, we couldn't really measure a very high activity from the trypsin inhibitors to begin with, because we are already working with a product that has naturally relatively low trypsin inhibitor activity compared to, for example, soy. But we did, so of course, in the mild fractions, that's also something that I should say, with the mild fractions, the native or without any heat treatment, we do keep the trypsin inhibitor activity. That is for sure true. However, with the processing step, the post-fractionation processing step that we did, this trypsin inhibitor activity went down to the same levels as the pea protein isolate. So with this post-fractionation processing, we can for sure reduce the activity. We did find something interesting in regards to the soluble and the non-soluble protein fraction. Trypsin inhibitors are usually, or well, they are smaller proteins compared to the big globulins or something like that from the storage proteins. So this where actually we could actually fractionate them into the soluble phase. We did measure a higher trypsin inhibitor activity in the soluble protein fraction compared to the non-soluble protein fraction. So this is something, yeah, I don't know if for some application this might be interesting, that you could actually separate them without any heat treatment. I don't know why that would be useful, but at least that's something we learned, that they can be separated into the soluble phase. So just by centrifuging the two, yeah, the protein-rich fraction. So that was something actually interesting that we saw, that the protein-rich fraction had the non-soluble protein-rich fraction did have low levels without any heat treatment, just because we could actually separate them into the soluble protein fraction.
Okay, thank you so much, Andrea. Shannon, do you have any question?
Yeah,
Are we having still time?
I have a question. Just one question. Thank you so much, Andrea. It was a very interesting talk. I really enjoyed listening to it. And I would just like to zoom back into your life cycle assessment. And I think most of us are aware of the new Delta model from the I think, sustainable nutrition initiative from New Zealand. I don't know if you're aware of it. So I just wanted to ask, what are the differences, if you are aware of it, that is, between your approach in your life cycle assessment and what is included in the Delta model?
Yeah, I don't think I am very well informed to tell you much about it. I should say here that this work that I presented with the life cycle assessment, it was finally kind of a culmination of two projects. On the one side, we had some colleagues that were working on the life cycle assessment, Anouk Lipiang, so you can find most of the data that we used in our work comes from her study on mild aqueous fractionation of P and lupin, if you're interested in that. So yeah, Anouk Lipiang did that. And then this work that I presented to you was done in collaboration with Nienke Dreyer. She was the one, this is that we came up with this sort of combined approach. So they were coming on from the side of the life cycle assessment, and I was coming from the side of digestion. So I have to say that here I think that this might be a better question to ask them. Maybe I can put you two in contact. Yeah, so for me, from my side, it was more the digestion part, definitely. Yeah.
All right, no, that's fine. Thank you so much for your response. Eva?
Yeah, I think that we are done with the Q&A section. Maybe we are receiving more questions. We will send it to you and Andrea. And now, again, thank you very much, Andrea. Now I'm sharing my screen. We have. So we wanted to take the opportunity to this webinar to invite recently graduated PhDs and people almost finishing the PhD related on in vitro digestion. As we are inviting these people to be a speaker on our webinar. We have available for November 1st and December 6th. So if you are interested, just reach us out. And also, we are in about to be. I mean, I think that the deadlines. The date for sending the abstract for our next Infogest conference on food digestion is soon. So for now, just we can say save the date. Our next conference is next year in April, 9 to 11 of April. And just keep in mind that the deadline for the oral and poster submission is going to be the 15th of December. And also we are taking the opportunity of this forum that we have the opportunity. So we are invite all of you to attend this World Food Forum of the FAO. So the title of this side event is Sustainability of Food Proteins, What Digestion Matters. This event is going to be advertising in our Infogest social media accounts. So have a look at it, and we will be happy to have all the people attending to this event. So the take-home message of this event, the first one is the quality of protein is a key metric that should be considered in determining sustainability of food protein sources. The second is that the in vitro digestion model are crucial tools for evaluating protein quality. And also, one of the most important is that younger generation are driving the shift towards sustainable and diverse protein sources. So please have a look at this, keep in mind, and we will be very happy to have you all in this event. Okay, I think that's all. So see you then in our next webinar in November.
Yeah.
Thank you, Andrea, again.